some of the routine clots that we encounter in our vascular models. For example, this one here is a soft clot that occludes in a bifurcation occlusion, very pretty simple kind of clot. The next clot we have is a softer clot that's a little bit more friable. It breaks up multiple times before it occludes in a very similar way to the previous one. Uh, and then here we have a clot that is really, really firm and occludes in the M1 and doesn't really move beyond that. And then we have some more heterogeneous clots, and this clot is a combination of a soft and firm clot. In this video, there's a prototype device that's trying to remove the entire clot, but you can see that only part of the clot actually gets removed, the softer part. And the same clot is then attempted with an aspiration catheter, where part of the clot is taken back, but eventually loses it. I think the interesting part about this in the last video is not so much the technique, but the fact that the heterogenic clot breaks up and that it can, in an angiograph, this one and the previous one can look exactly the same, because the occlusion, you can only see the proximal occlusion. You can't see what happens more distally. So the whole reason for showing some of these clot videos is because I think that there is quite a complexity in clots out there. And so in trying to remove it, probably also requires complex thinking. Just a summary of all of the clinical trials that have gone so far, doesn't include Dawn right now, but you can see a very nice, steady increasing trend of clinical outcomes and reperfusion in blue and clinical outcome in green. But there still remains a gap between getting better reperfusion and also what's often referred to as the futile recanalizations, closing the gap between reperfusion success and good clinical outcomes. The Hermes analysis that was conducted, the meta-analysis that was conducted, shows still approximately 30% of occlusions are not reperfused. And there's a lot more evidence now. For example, this paper that was looking at Tiki 3 versus Tiki 2B to suggest that that complete reperfusion is a lot better than partial 2B. And this, what I did was go through all of the mechanical thrombectomy publications in 2016 and selected those ones that reported 2B and 3 separately. And this shows that there's still that roughly half of the population, a little bit less than half of the population, is completely reperfused. So suggesting that there is a significant amount that can still be improved. And so all of this suggests that there are certain challenges to clot retrieval. Again, can be broken into those two gaps as what's often referred to as futile recanalization or failed recanalization. And there could be patient selection or procedural criteria both involved on determining the success of a futile recanalization and also from a failed recanalization perspective. But I think what's notable is that there are significant clot and so from my perspective, or our perspective as a company who is looking to design a device, for example, at the complexity of stroke, we really wanted to make a bench model that was capturing all of these types of challenges in stroke so that we could design a device. So really, we went through the effort of not only looking at all of the anatomical variations that exist and trying to model those, but also the clots that cause the stroke. So we developed population-based vascular models and also population-based clot analogs that represented the clots encountered in human stroke population. And what this allowed us to do is then to use these models for investigating different prototypes before we arrived at a device that we thought was good. in this process, we learned a whole lot about clots and their challenges. And this talk is specifically about some of those learnings that we had. To start off with, there are now quite a few good publications out there that have taken clots from stroke patients and analyzed their composition using quantitative histology. And these three publications do it in a very similar manner. So what we did was we grouped the results and made one chart that represented 100 different clots from 100 different patients, and it's just and ordered them by composition. So what's interesting here is the enormous variation in clot composition that exists from stroke patients. Clots that can be up to 80% red cells and very little fibrin meshwork, and on the opposite end, clots that are densely fibrin with almost no red cells incorporated. And so using this composition data and different mechanisms of making clots through through Chandler loop, for example, that might simulate a high shear stress environment or more static environments by adding different elements like ADP, thrombin to make the clots different. We were able to replicate clot analogs that were really representative of the human clot compositions. And so this publication that from our lab shows that the clots that we make from animal blood have a very similar composition, and we can do them repeatedly to the clots in humans. Of course, they're in vitro, and most of them are homogeneous, but they're very advantageous to us because we can then make them repeatedly the same again and again, and then test their properties and really understand why some behave the way they do. This is just an example of some of the clots that we can make. We can really make over hundreds of different types of clots. We have protocols that allow us to do that for loads of different composition ranges. But the publication that I showed in the last slide really narrows them down to 7 different clot types that are very different from one another. But of course, looking at these clots really begs the question, if they look so different, do they also behave very differently? So I want to start again with another video that shows a prototype stent retriever trying to retrieve 2 different clot types. The first on the top is kind of the middle of the range clot, about 35% hematocrit from a sheep blood, and the bottom one is about 95% fibrin or plasma incorporated in there. And this stent retriever in the top video can really engages the clot, pulls it back very nicely. Really nothing to see here. You know, this is normal, or what you hope is normal. And in the bottom video, then this gets very interesting, where the clot is so stiff and firm that you can see the stent retriever collapse, doesn't really engage with the clot. The clot doesn't move, and it kind of rolls at the end over here. And fails from a retrieval perspective. And when we look at the images, the before and after images, we can notice that, of course, is not removed, but also it seems a little compressed. And this coincidental marker shows that it's flipped. So it's really gone through a lot of changes. And as engineers, what we really want to do is quantify those changes so that we can truly understand their behavior and design a way around it. So we've really investigated a lot of their properties. I just want to talk a couple of those properties here today. The first one being the property of friction. So in the background to this is that in an occlusion, there are pressures that direct the clot forward, and there's a net gradient pressure that goes distally. And the clot, being in the vessel, encounters friction, but probably also biological adhesion between the vessel wall, and that all has to be overcome to be able to retrieve. And so this is why we were interested in studying friction, is because it's a really important force that if we can quantify, will help us to know how to retrieve it. So in order to test this, we really created a simple kind of crude test methodology where the clots are placed on top of a slide, and that slide is inclined at an angle, and the tangent of the angle when it begins to slide down gives us the coefficient of friction. So you may have seen this video from the journal Stroke publication, but we tested it on different clot types and different substrates, including bovine aorta. And we were then able to find the polymer substrate that had similar results. And so this video shows on the left a red cell rich and the right a fibrin rich. And you can see how quickly this red cell rich clot slides down, but the fibrin rich clot is very, very stubborn, doesn't really move. And what's very interesting is not just that there's a difference, but that there's a huge difference. And of course, these two clots represent the extreme ends of the composition. So what we did was we redid the study with a whole load of different compositions to find out their effect on the coefficient of friction. And last year we published on this to show that there are certain clots, those that have a really low red cell volume, or vice versa, a really high fibrin content, that have a significantly higher coefficient of friction, suggesting that inherently these clots can be very difficult to retrieve because of their inherent properties. So when you saw the first video of the device trying to retrieve it and failing, and we saw the clot was compressed. So what we wanted to do now was simulate that compression again. So we took these clots and we compressed them on an on a surface, and then re-performed the friction study to see how much effect the compression then has on the friction. And again, what we found was that compression of these clots further increases the coefficient of friction. So this suggests that the best pass is probably the first pass to be able to remove, and that each pass will subsequently modify the clot property. And on the other end of the spectrum, we also have looked at these challenging red cell rich, soft, mushy clots that that tend to break up really easily. And in the videos here, what you can see is that there's two mechanisms: aspiration and stent retriever with a catheter. And in the aspiration video on the right, you can see how the clot breaks up because it's not controlled. And in the left, the clot breaks up because it encounters shear force with the catheter. So this video is not to show one technique over another, but to show that clots inherently, some of these, can be very friable. And again, if we want to try and understand their quantification a bit more, we looked at their resistance to indentation to understand this purpose. So we again tested with a lot of different blood compositions of clots, and put them in this indentation test machine over here, where a clot, you can see on the right that little red box. Inside the red box, the clot goes inside there. It's surrounded on all sides, so there's only the pin at the top that can really push down. And in the middle picture, you can see the middle right picture, you can see the clot indenting by this pin. And the pin was designed as a same dimensions as the strut of the device. So to try and see how the device will engage into the clot. And it's being pushed at the same, or maybe not the same, but the similar force of radial expansion of the device. And so after this indentation, over time, we took each clot out and looked at it also at the indentation site using scanning electron microscopy. So what we first observed was that initially, in the first minute, all clots showed a very similar indentation depth. So the fibrin-rich clots as well as the red cell-rich clots kind of show similar indentation depth in the first minute. So the interesting observation happened after the first minute. And this graph over here shows the incubation time over 5 minutes for each clot type. On the left is a 0% red cell, so it's 100% fibrin. And there's two of them because the high density one is actually compressed and the low density one is not compressed. So it's the left one simulates a failed retriever. And the right one simulates just a fibrin-rich clot, and then there's a 5% and a 40%. And what's very interesting here is the creeping incubation time of the 0% clots that continue to embed each minute, whereas in the red cell rich clot, it kind of plateaus a lot more quickly, as you can see on the graph on the furthest right. So this is suggesting that these fibrin-rich clots continue to provide resistance towards indentation, and to embed them more completely probably requires more time. But the red cell rich clots indent very quickly in the first minute and plateau over the 5 minutes. So probably don't require that amount. But looking at the SEM images further revealed how the fibrin meshwork was behaving. So on the top are the baseline images, and in the middle are the indented samples. And if we zoom into the indented samples of the 0% and the 5%, which are both representing really fibrin-rich clots, we can see stretching of fibrin fibers. So the continued indentation causes the fibrin fibers to first align and then stretch, and we can see this plastic deformation after the 5 minutes are over. And in the red cell rich clots, there is some stretching, but also we observed fibrin rupture in some of these clots. So this probably suggests why some of these red cell rich clots can break up more easily and embolize. Um, we've also tested a little bit in vitro on the impact of TPA. So really, what we wanted to do was how TPA affects the composition and then further the microstructure. This is not a complete study, but part of it. So what we did was we created a flow model in which we circulated human plasma at 37 degrees Celsius. Clots were inputted into the MCA. We put a clinical dose of RTPA, incubated it over 2 hours, and then we measured the degradation of the clot over that time period. And after that, we fixed the clot for SEM analysis. And what we found was that sometimes this feels a bit counterintuitive, but it is very explainable that the red cell rich clots break up a lot, and so you can see they are the ones that degrade most, whereas the fibrin rich clots degrade the least, probably because even with circulating plasma, the TPA is only encountered on the proximal side of the clot, and there's such a dense fibrin meshwork for it to penetrate into that it does degrade the TPA, or it does degrade the fibrin, but clearly not as much. And with the red cell clot, you only need a small amount of TPA to break up those fibrin fibers, because there are only a small amount of fibrin fibers to begin with. And then this kind of got confirmed after we looked at after the images of both of these clots using SEM. So the top one is a fibrin rich, and the bottom one is the red cell rich. And you can see it's not really zoomed in, but there are fibrin ruptures in the small box on the bottom right, and actually in the top right there are also some fibrin ruptures because of the TPA, but due to the dense fibrin meshwork in the top clot, it just doesn't break up. So all of this research that we do, sometimes we get asked, why do you do all of this from a company perspective? But we do this research because we believe that it helps us to understand these specific challenges, and then to be able to design around with a device. But also then we can test with different procedural techniques. So just a couple of slides to show the idea that we can, under our flow models, use real clots, real devices in very real settings, and really understand how clots interact with devices, and then also test different procedural techniques, like the one shown over here, sometimes referred to as the epic or the wedging technique, where a clot is partially retrieved by the device and then pulled back. And also to look at flow dynamics. So we have the ability to investigate how different how flow changes with different techniques, different clots, different devices, and to really look at that behavior. So, really, in conclusion from this presentation, I think we've really identified that there are different clots presented in human stroke, and they do present different challenges. So, in the middle range of the clot composition, 50% of the clots that are soft but also quite cohesive, they don't break up so easily, they're probably going to be retrieved by almost any system. But the challenges really exist on the periphery, those clots that are really easy to break up, and those clots that are really, really firm and fibrin rich. So we think that better understanding of these clots can really lead to better solutions from a device design and a technique perspective also, and that in vitro modeling, it can be valuable, but really the output of modeling is only as good as the input. And this is why we spent a lot of time having really good inputs like these clots to give us appropriate outputs. By no means is this complete. There is so much more to learn